food, drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info. Bement, thank you for joining me.
Yeah, it's great to be here. And I go by Bill.
Okay. Yeah, I remember that. For those listening, fun factoid. So Bill Bement is a cell biology professor at the University of Wisconsin, where I went to college, and I took his course many, many years ago, probably like 12 years ago or so now. And so anyways, can you tell people a little bit about who you are, what your lab studies, and what you think about?
Yeah, so I've been a professor at the University of Wisconsin-Madison for a little more than 30 years now. And our lab has been studying for that time what we think of as patterning of the cell cortex. The cell cortex being the outermost part of the cell, which would include the plasma membrane and the stuff immediately beneath it. And we study 2 processes in particular. One is cell division and the other is cell repair.
Mm-hmm. So cell cortex, like I, as a neuroscientist, and someone who talks to a lot of neuroscientists, this cortex of the brain is the outer part of the brain, the wrinkly part. The cell cortex is then, so the outer part of a cell.
Yeah. So the cortex is apparently an old term. So you'll run across it in many different contexts in biology. Trees, for example, have cortexes, brains, as you know, and cell cortex is just the outermost part of the cell. So I guess in general, when you hear the term cortex, think outer part. So for the cell, that would be the part of the cell that bounds it. It forms the boundary of the cell, divides the inside from the outside, and it's a remarkable structure insofar as it receives information from both the outside of the cell and the inside of the cell, and it responds to it in a fairly sophisticated way. So it can transmit information from the inside to the outside of the cell. It can interpret information. It can actually transmit information from one part of itself to another part. So I
Mm-hmm.
think there's this tendency for people who talk about cells to think of the nucleus as being the brains or something like that. But I think that a lot of people would argue the nucleus is just sort of a repository of information, and other parts of the cell, including the cortex, that actually have the job of interpreting and thinking, as it were.
Doing the computing, if you will.
Yeah, at least to a certain extent.
Yeah, yeah. Interesting. So the cell cortex, is that, should we think of that as being synonymous with the lipid membrane, or is it lipid membrane plus something else?
Yeah, the second of those two. So it's the membrane plus the proteins that span the membrane, and then the proteins that are just beneath the membrane. So that it's very dynamic, but you'll have typically proteins that are coming from the interior of the cell or the cytoplasm to the plasma membrane reversibly. Okay, so its makeup is constantly changing, but what's typical for the cortex, besides comprising the membrane, is a lot of cytoskeletal proteins like
Mm.
actin, filaments, and myosin, the things that power cell shape change.
Mm-hmm. And when we think about the cytoskeleton, if I kind of think about the history of my knowledge of the cytoskeleton, and when people start to learn about this, I think where it often ends is they think, okay, cytoskeleton, there's some little fibers and cells that maybe give them their shape. They're involved in cell division. So when a cell has to divide and chromosomes have to get pulled apart and all that stuff, the cytoskeleton is involved in that. But there's a lot more to it than that. And I really don't know that much about it. But when we start thinking about the cortex, like, can you start to give us a sense for let's just start with the major pieces of cytoskeleton. What is actin? What are microtubules? What are some of the major players that we might be talking about today?
Yeah, so actin is a protein polymer. It forms long filaments, and the filaments can exert force in a couple of ways. One is by growing, they can push on things. Okay. And a good example of that would be when cells are crawling around. There's typically a lot of actin filaments growing in the part of the cell that's extending forward. But they can also produce force in collaboration with a motor protein called myosin, and in particular, the one that's most commonly associated with the cortex is called myosin 2. And it's actually in many ways similar to the protein that powers contraction of our muscle. There are some differences, but to a first approximation, it really is a lot like the proteins that make our muscles move. It just happens to be in essentially all cells in our body, helping the actin power shape change by contraction.
Mm-hmm. So you've got these little filaments, we call them actin filaments, and they can grow and expand, and they're like little fibers that can, push a cell forward. So if I imagine something like a white blood cell that has to move around a lot, it's kind of feeling around and sensing things. The actin will have something to do with the physical motion of that cell.
Yeah, that's exactly right. So saying something like, oh, the actin and the myosin power cell shape change is accurate, but that encompasses a lot of different kinds of shape change. So there's the ones you're alluding to, a white blood cell moving toward a bacterium.
Mm-hmm.
That requires a lot of very complex cell shape changes to both move toward the bacterium, and then when it gets to it, to engulf it so it can destroy it. As you alluded to, cell shape change also means the process, or also comes when you split the cell in half at the end of mitosis. It happens very commonly during development when tissues get reshaped to form the complex embryo and then eventually the adult. So, and then there are all sorts of interior shape changes that are maybe a little bit more subtle, organelles getting changing their morphology or being transported.
Mm-hmm. Mm-hmm. And so these
All
These
due to this.
myosin proteins, can you just give us a basic sense for how are they interacting with actin? Are they physically grabbing onto it? Are they moving along it? How do they tie into the biomechanics there?
Yeah, that's a really interesting question. So the myosin binds to both actin filaments and to ATP, the energy source of the cell or the major energy source. And the idea is that the process of binding to the actin triggers with some regulation, a cycle of ATP hydrolysis that's coupled to movement of the myosin. So it changes its conformation. And that conformational change is coupled to movement of the actin. And it can work the other way too. If the actin is anchored, myosins can move along the actin filaments. But again, as a consequence of this ATP binding analysis cycle.
So this would maybe be a good example of a time and a place in the cell when the ATP, the energy currents of cells, is actually being cashed in to do something. It's actually driving physical changes that, in this case, are shaping actin filaments and moving cells around or changing their shape.
Yeah, exactly. I think it is one of the easier things to think about in terms of energy utilization by cells because we're all familiar with motors, and that's exactly what we're talking about here.
Hmm.
Myosin is considered a motor. Um,
Yeah, yeah, yeah. And so, you mentioned this, but these things are all dynamic. So the actin filaments are not static. They don't sit in one place. They're not a fixed length. They grow, they get shorter. This makes sense if a cell has to move around and change shape, but they can also be anchored. Can you just sort of give us a sense just for how dynamic these things are? Can they kind of be stuck in place to maintain shape? I would imagine the answer is yes to some extent. Are they moving around like constantly nonstop, or is it really just a mix of all of the above?
a, a mix of all the above. So depending on the cell type, the actin filaments can be pretty stable or astonishingly dynamic. So I didn't mention this before, but the actin monomers, the parts that make up the filament, bind to ATP as well, and they can also hydrolyze ATP, that is, utilize the energy stored in the ATP. So that process comes with actin assembly and disassembly. So the actin filaments are growing, they're shrinking in a relatively stable situation, as you might find in muscle, because you don't want the actin filaments to be turning over too quickly. There, the turnover is kind of modest. I think the general idea is you replace the filaments as they get old, but otherwise, you want them to be there so you can rapidly contract the muscle when it needs to be contracted. But in something like the white blood cell you mentioned, there the actin is turning over very, very quickly. So it's not only getting pulled on by the myosin, it's assembling and disassembling. And this is all coordinated to result in net movement of the cell from one place to another.
So depending on the cell, depending on the conditions, these things can be relatively stable and they can be extremely dynamic. When we think about how they change in their shape, in their length, is this is what's is an actin filament like a string of Lego blocks that I can add and subtract blocks from the ends, or does it have more thickness to it? Can you give us a sense for like how it grows and changes?
Yeah, so it's helical
I see.
and grows by the addition of the subunits, also known as actin monomers, to a specific end usually. So one end grows faster than the other end.
Hmm.
And that is made possible by the ATP hydrolysis. The kinetics are kind of complicated, but it's very typical for cytoskeletal proteins to have two distinct ends, or the polymers, that is. So picture monomers floating around in the cytosol, and then they have the opportunity to add to the rapidly growing end of the filament. And if that's occurring, that makes the filament grow. There are other proteins that can facilitate that process. That is, they can associate with the actin filament and then accelerate the addition of monomers to one end. There's a lot of regulation. There are also proteins that can bind to the slower growing end to protect it from disassembly. There are proteins that can bind to the fast growing end to protect it from disassembly. And then there are proteins that will sever the filaments.
I see. So actin filaments, they're filaments. They're relatively thin. They can get pretty long. They can be very dynamic, but you can kind of plug them or stop them by attaching stuff to either end to keep it relatively fixed. You can anchor it in certain places if the cell needs to, and it's helical. So it's not like it's not like a street light post. It's more like, I don't know, like a corkscrew shape or something like that.
Yeah, not without quite such a dramatic twist.
Yeah.
You also asked about or I think you were wondering about the relative stiffness. So compared to some polymers, it's not very stiff. So it does have the potential to bend relatively sharply, and that contrasts with something like a microtubule, which can bend, but they tend to be a little more stiff,
Mhm.
and they more stiff. Um,
Yeah. And so, what are microtubules in contrast to actin filaments? Are they just sort of like bigger, thicker, stiffer structural components, or?
They're definitely bigger. They are, as the name implies, tubes. So they're hollow tubes. They're also assembled, though, from smaller subunits. In this case, dimers of the protein called tubulin. So they're roughly 3 times the diameter of an actin filament, or 3 or 4, depending on how you do the measurement. And that means in turn that they've got a lot more subunit because the size of the subunits aren't really that different. An actin monomer is about 42 kilodaltons, which is a standard measure of size for proteins. And the proteins that make up the two parts of the tubulin dimer are about 55 kilodaltons. Okay, so you can pack a lot of those into a microtubule. And on average, they tend to be stiffer if you're talking about a single microtubule versus a single actin filament.
Okay, so actin filaments are thinner. They're made of smaller pieces, basically. This is not like a steel beam in a building that's completely hard and stiff. It's got a good amount of flexibility and bendiness to it, maybe. And that would kind of make sense if I think about a cell very, very intuitively, right? They're not stiff like Lego blocks. They've got some give to them. They can sort of reach out and have protrusions and all that stuff. Microtubules are bigger, and as the name implies, they are tubes. So there is an empty space inside of them.
Yeah, there is. It's becoming apparent that there are maybe other proteins that are found in that empty space. So it's perhaps not as empty as has been traditionally thought, but it is nonetheless a tube, as opposed to, the actin filament, which would be a little more analogous to, say, a DNA double helix. So
Hmm.
two
Yeah.
two complementary strands wrapping around each other. Um,
Yeah, yeah. So these actin filaments, yeah, they're constantly growing and changing to some extent. They have that helical structure. We'll probably talk a lot about those microtubules. I just want to ask, tubes. What do we use tubes for in our houses and our buildings? Well, we put stuff into the tube, and whether it's the stuff going through the toilet or the water in the building, whatever it may be. Are the microtubules purely structural in a skeletal sense, or does stuff flow through the tubes?
I don't think there's no evidence that stuff actually flows through the tubes, and it's a very small diameter, so something like 15 nanometers. Moreover, if there are proteins in there, essentially they're going to be narrowing the lumen, as it's called, even further. So it seems unlikely that there's actually going to be anything of size moving through the tubes. Um, Something small, in principle, could move through, but it would have to be quite small, especially if the tube is any length. So they have, depending on the cell type and the organism, the microtubules both they can serve as structural elements. So red blood cells in mammals is a great example. They form a circular array around the perimeter that helps keep the red blood cell, sort of disc shaped. But in the typical cell, they're possibly best known for separating the chromosomes in mitosis. So they also have their own associated motor proteins. And they also have this mysterious impact on the actin and myosin. So they serve a regulatory purpose. And this is most evident during cell division when the mitotic spindle, which is made up of the microtubules in the chromosomes, sends signals that essentially tell the actin and the myosin where to go, so that the actin and the myosin can split the cell in half.
And, we might look at some movies here today. And one of the things I remember, I believe when I took your cell biology course back in the day, there were like these new videos out and they were, just like computer animations of all of the stuff happening in cells. And, among other cool things you see when you watch these movies is you see these filaments become assembled and disassembled. And what you notice, at least what I remember noticing, is it's not like there's a little protein with hands in there that's piecing them together one at a time. These things just sort of come together magically, and then they can kind of dissolve magically. So the filaments are self-assembling somehow in some way. At least that's how it looked in that video that someone made for didactic purposes. Can you talk a little bit about how these things get put together?
Yeah, so there are two ways to think about it. One is what happens with the purified proteins. So using actin as an example, if you have actin monomer bound to ATP above some threshold concentration, it will spontaneously self-assemble into a filament. And the same is true for tubulin. So strictly speaking, neither of them needs help as long as the subunits are above a critical concentration, need help to assemble. These processes, though, tend to be very heavily regulated in the cell because the cell doesn't want the polymers to be forming, except really where they're supposed to be forming. So it has mechanisms to prevent them from forming where they're not supposed to, and then other mechanisms to promote their formation where they are supposed to be. So, yeah, otherwise you're right. They will spontaneously fit together to form their characteristic structures, actin filaments in the case of the actin and microtubules in the case of the tubulin.
So
And tubulin, I should mention, also has a nucleotide, a GTP though, instead of ATP associated.
So the actin monomers, the units of the actin filaments, they bind ATP. If they are around, if they are bound to one another and present above a certain concentration, they just start stitching together into this filamentous shape. And I don't know, can we just think about it as like, two magnets stitching together? Is this all like electrostatically driven? Is it, how does that happen?
Yeah, I'm trying to think. You mentioned the Lego, and imagine you instead of just stacking the Legos on top of each other, imagine you have a brick here, and then another brick can associate with the side of it, but with some overlap. Okay, so now that creates a little nook into which a new brick can fit. That is the spot where there's overlap, if you get what I'm saying.
Mm-hmm.
And that will, of course, generate a new nook into which a new one can fit. So the conformations of the proteins are important, as are the charge distributions. So tubulin's a little more complicated because it assembles in a sort of, there's some debate about this, but it because it forms a tube, it's presumptively a little more complex when it assembles. And some of the best data suggests that what happens is it starts at the fast growing end as a sort of sheet that then curls up and zippers together. But in any case, the protein conformations are important, as are the distributions, which go in hand in hand with, the charge distributions, which go in hand with each other.
Mm-hmm. And so
there's one other point that I worry that I may not have made, but which is extremely important. So both cases are associated with a nucleotide. So ATP for actin, GTP for tubulin. And in both cases, the assembly process is associated with a hydrolysis of the nucleotide. So it will go, in the case of actin, from ATP to ATP. And you might assume that this provides the energy needed to build these structures, but in fact, that's not true. You can basically get an actin filament even with a modified form of ATP that can't undergo hydrolysis. And the same is true for tubulin and GTP. So that it's not the case that the assembly of these structures is being powered by energy provided
Mhm,
by
mhm.
nuclear
And
hydrolysis.
can you just define hydrolysis for people that don't know biochemistry here?
Yeah, very simply, the something like ATP has different form, or so ATP has three phosphates, and the one of the phosphates is typically broken. So the between the second and the third, there's a bond, and when that's broken, that releases energy. So that comes with the conversion of the ATP to ADP. And when people talk about ATP being used as an energy source, that's usually what they mean. The ATP is getting converted to ADP. And for GTP, it's essentially the same thing.
Mhm.
The ATP is getting converted to
Yeah.
GDP.
But what's the involvement of water there?
Water is essential to break the bond, but we tend to think in terms of what will happen spontaneously and what won't. And because
Mm,
they're in a normal, happy, healthy cell, there's plenty of water around, you tend to ignore it. Same with certain ions. So the reaction
yeah.
also requires like magnesium, but there's usually plenty of magnesium in the cell as well. For better or worse, we focus more on
Yeah. Yeah.
what are the things that will make the hydrolysis more efficient or faster.
Yeah. So when we think about ATP and energy, this ATP, three P's triphosphate, the a lot of the energy that's converted into kinetic motion in this case, when we think about like myosin and actin and stuff, that is coming from sort of clipping one of those bonds, and that's releasing the energy.
Yeah.
And so thinking about the cell cortex some more, so you said it comprises the lipid membrane itself plus some. So is there generally like a bunch of actin in some kind of skeletal structure or pattern or shape just beneath the cell membrane? And we would include that in what we call the cortex?
Yeah, that's exactly right. So the so-called cortical cytoskeleton is a layer of actin filaments and myosin that is present in most cells, and it's just beneath the plasma membrane, and it's physically linked to the plasma membrane by a variety of different proteins, some of which span the plasma membrane, and others of which sort of bind to the inner surface of the plasma membrane.
Mm-hmm. And I would imagine, right, the details of all that are important for how fluid things are in the membrane, how locked in receptors or whatever are into particular spots on the cell, and all of those types of things.
Yeah, that's quite correct. You in general, the cell wants things to be reversible, um, and so everything tends to be reversible. And then we draw a distinction between tight binding and loose binding. And tight binding simply means that two things spend more of their time associated with each other. So say you're in a cell like I mentioned before, the red blood cells in our bodies. You're going to want that microtubule bundle that defines or helps shape the disk of the red blood cell to be anchored pretty firmly to the plasma membrane. So you want tight binding. So there, the interactions will probably be tighter than cases where you're going to want rapid cell shape changes, like in a white blood cell, which doesn't maintain a steady shape, but instead is very protean and it undergoes changes constantly.
Mm-hmm. And when we think about sort of the concept of self-assembly or self-organization, can you talk a little bit more about what this is and what drives it? So you mentioned that, okay, we've got actin filaments, they're constantly growing and assembling and disassembling at different rates in different places. This has something to do with the concentration of things like the actin monomers themselves, coupled to these pieces of ATP. Is it all, just I'll have you just sort of define self-assembly or self-organization. And is this all concentration dependent, or what are the other factors that are, is it pH dependent? How much of it ties into things like electric charge? What are the factors here that need to be present that change that dictate whether or not something can self-assemble like actin?
Yeah. So people distinguish between self-assembly and self-organization.
Mm,
And we'll start with self-assembly because we were just talking about that. So self-assembly does not, strictly speaking, require energy input. So actin and tubulin, going to actin filaments and microtubules respectively, would be examples of that. Although, as we said, they're associated with nucleotides, they do not actually have to hydrolyze the nucleotides to assemble. So, other examples would be that you're probably familiar with things like a lot of viruses will self-assemble if you give them, if you have all the components, they'll form
yeah,
fairly
yeah.
like structures. Strictly speaking, that does not require energy as long as the proteins are above some minimum
Hmm.
concentration.
They're just going to all kind of snap into place.
Yeah, exactly. It's a little eerie too. If
Right.
you now have the capacity to watch it, maybe not at the quite at the level of the individual things, but you can see filaments coming apparently out of nothing. If you have a mixture of actin and it's above some concentration, you start your movie and it's fluorescent. It's like there's nothing, and then whoop, here comes a filament. Same for
Yeah.
microtubule. Um, Self-organization is a little more complicated. It does require energy by definition, and it typically refers to higher level processes. So if you have mixtures of, say, actin filaments and myosin, or microtubules and various microtubule binding proteins, they can self-organize into more complex structures. And this is an example of something I think you've covered before in your podcast. But basically, there are emergent behaviors that characterize self-organization. So put really simply, the output of a mixture of things is not predictable based on the individual components. You have these sometimes even more eerie and more remarkable things forming as a result of self-organization,
Hmm.
but it comes at the expense of constant energy investment.
Hmm. Hmm. So self-organization can stop if the energy stops, but self-assembly can just sort of happen automatically, if you will, because the pieces are just going to come together without any energy needing to move them together.
Yeah, with the caveat that we're talking about with the second law of thermodynamics tells us that you're not even if something like a filament can form spontaneously, it's not going to last forever.
Hmm. Hmm.
Uh, but yeah, otherwise you're exactly right.
Okay. So actin filaments, they self-assemble. So this is self-assembly. It doesn't actually require energy input to happen. Very concentration dependent. As soon as you get enough of the pieces together in a high enough density, this just tends to start to happen. These filaments form. It even looks eerie, and maybe we'll see some movies of that. Self-organization. What would be an example of that? A common example at the cellular level of something that's a self-organization and not just a self-assembly.
Yeah, the how long has it been since you looked at a mitotic spindle? And be honest, it was,
It,
when you took the cell biology class, what was it, 15 years ago
yeah,
or
well, looked at one. Yeah, I don't know if I've looked at one since then.
so you probably remember at least that that there's the chromosomes are lined up in the middle of this thing, and there's a sort of spindle shaped array of microtubules that contact those microtubules and then extend outward away from it. And those then come to sharp points, hence the name spindle. Although nowadays I feel like how many people actually know what a spindle is? Probably not many,
Probably
because
not many. Yeah.
um, uh, we'll say it's roughly football shaped,
Hmm. Hmm.
maybe a skinny football with the chromosomes lined up in the middle. And then the microtubules come to focuses at either end at some distance from this chromosome array. And then they also have microtubules sort of sticking out away from the chromosomes. Okay, so it looks very complicated. That is nonetheless an example of self-organization, and some truly marvelous experiments done by Rebecca Hield um, back in the mid 90s show that you can take DNA coded beads and then put them in a cell extract, just beads coated with DNA from viruses, so not chromosomes. Okay, just DNA on beads. You can throw that in a cell extract, and under the right conditions, something that looks exactly like a spindle will form. The
Hmm.
microtubule will contact the beads in a, sort of a football shape. And if you didn't know better, and in fact, the chromosomes are round, or the beads are
Right,
round. So,
right, right.
so that's an example of self-organization.
I
And
see.
at the time it was done, it was completely mind-boggling because everyone assumed that there was a lot of top-down control over spindle
Right,
assembly.
right. I was about to say, yeah, you might think naively that that level of organization is put in place. Like there's like a little, a little demon in the cell that needs to like, try to arrange it a certain way. But what we're saying is this stuff happens automatically in, you use the word eerie a couple times, and you could even put a bead, which is a sphere, right? It's not even the shape of a chromosome. You just put DNA on a bead, and as long as like the right stuff is in the soup of the
Yeah,
experiment, like this thing self-organizes. It just happens.
yeah, it's depending on your perspective, either really, really cool or deeply disturbing. I tend to think it's really cool in the sense that it makes you realize that maybe a lot of the things we assume are quite complicated are actually the outcome of a bunch of simple rules.
Yeah.
Uh, and when repeated iteratively, those rules can give rise to fairly complicated looking things.
Yeah, yeah, yeah. I don't know how good this analogy is, but I right now I can't remember even what context I saw this in, but if you watch a video of like a trout, like a fish in a stream, and it looks like it's staying in place in the moving stream, you think, oh, the fish is, it's got a body and muscles and it's trying to stay there. But you can take a dead one, and it's just by the shape of it, it just kind of like stays and swims in place, even though it's dead. And
Okay.
yeah, like stuff like I guess the point I'm just getting to is like some of this stuff happens automatically. And when we're talking about a cell, so when I say the word try or I anthropomorphize, it just illustrates how difficult and eerie this stuff can be to talk about. But like some of the stuff just happens as a natural sort of biophysical, electrical consequence of how the pieces match up.
Yeah, that is exactly right. And the more we learn about the pieces, the more common this theme seems to be. It just sometimes it's a matter of doing the right experiment. Because you can see a behavior in an intact cell, an intact living cell, and it can be really, really, wow, that looks so cool and that's so complicated. How do they do that? But then with work, you have the potential to actually find that, oh, the as cool as that looks, as complicated as that looks, it's down to some behaviors that are actually not that, complex.
Yeah, yeah. One of the things I want to ask you about, and this might be kind of a vague question, I'll let you kind of direct us towards actual phenomena we can talk about. But, oftentimes in biology, when we talk about complexity and information flow and information encoding and patterns of stuff, we think about things like the nervous system, like where most of my studies took place. It's not too difficult even for a non-scientist to think about, like, okay, our brain has many neurons, they're connected to each other, they can each send a signal, and I can imagine one neuron sending a signal to another, and then another, and then another, and then there's going to be patterns of this stuff in this network of neurons. But there's lots of information and patterning and flow of energy and stuff even within single cells that happens. They're not just sort of like these inert little blobs that are, fully static. When we start to talk about things like patterning the plasma membrane or waves of enzyme activity within a cell, what are some good examples of these sort of within cell patterning events that happen? And I want to start to talk about some of the dynamics and how that happens.
Yeah, so focusing on the cortex, which is what we put most of our effort in, the 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 control proteins for actin and myosin, which are generically referred to as the Rho GTPases, they characteristically form really interesting and striking patterns. And the idea is where you see an accumulation of these proteins, there will follow the accumulation of actin and myosin, and then that's where you're going to get the needed cell shape change. So, picture, for example, during cell division when the cell is about to split in half, a stripe-like pattern of waves of a particular protein called Rho that wraps around the cell, the cortex, around the equator. Okay, just like it sounds, the equator of the Earth, but much smaller. And then that's followed by the accumulation of actin and myosin in waves, and then they can do their thing. They can drive contraction around the equator, thereby splitting the cell in half. That one's relatively easy to understand. There's another example. If you wound a cell and the wound is big enough, you'll get rings of, again, Rho GTPases. In this case, they're concentric. So one called Rho itself will form a little ring around the wound, and it'll be circumscribed by another ring of a protein called CDC42, which is another small GTPase like Rho itself. And once those rings are established, then the cortex closes over the wound site. So those are pretty straightforward examples, but there are others, and um, a lot of them have been discovered pretty recently, just because the interest is rising in cortical behaviors, and then also the technology has become capable of detecting them. So, in other words, you need to know in some cases that you're looking for them, and you also need the
Tools to do it.
yeah, the imaging tools that make it possible.
Yeah. So imaging tools, like you can watch this stuff.
Yeah, they happen on a time scale that to our eyes is a little slow. So you typically use time-lapse imaging. But if you've got a time lapse imaging setup, if you have the capacity to follow the proteins in question in living cell, that's really all you need. If you're talking about wound healing, it's of course you need the means to wound the cell to watch the healing response unfold. But
Hmm.
a lot of these things just happen spontaneously. For example, many early embryos, mammals, frogs, flies, with the right tools to detect, say, actin, and again, the right image approaches, they spontaneously generate waves of actin that just move through the cortex constantly.
Hmm. And before we talk about some of the technologies and get into the wound healing stuff, did you have some videos where we could see this and just give people a sense for, we'll look at this at multiple levels, but you could watch these videos and have no idea what you're looking at, and I think probably what you're going to show us is going to be like, whoa, that looks cool, but then obviously we'll have you unpack what's going on, and then maybe talk about how the hell you guys actually put these videos together.
Sure, I'll see how this works. We, I'll share my screen, and
Yeah, there
depending
we
on
go.
the quality of our connection, you'll just have to tell me if it's playing. Okay, so we're looking at a cell, okay, and we have fluorescent probes, protein.
I see your cursor and I see black and it says cell wound healing.
Okay, the in the middle of the screen, we'll be able to see it or not. Can you see underneath the text that says active row in green and then active CC42 in red?
No.
Well, let me play the movie, and the movie's probably more important.
Okay.
So,
There we go. Oh, I see all of it now.
okay. So basically, in this experiment, we're looking at a cell, we start filming it, and then we fire a laser into the cell to create the wound, and then we watch the response as it happens.
Wow. So we're looking at one cell, and so the center of the ring that I'm looking at would be you've blasted a hole into it with a little laser.
That's exactly right. And cell repair is something that's becoming increasingly popular to study because it's apparent that certain diseases are underlain by deficits in the repair capacity, or in alternative cases, an excessive damage just happening spontaneously. So this is one way to study the process. You can induce a wound with a laser in this
Mhm.
case. But when you and I are sitting here just talking to each other, there's naturally damage
Right,
to
right.
um,
And so I mean, so at least simplistically, I'm looking at sort of the whole closing up here effectively.
yeah, that's exactly right. And the pattern we're talking about here is just the sort of concentric rings. Okay, so
Yeah,
we're
yeah.
looking at a cortical pattern of a protein called CDC42 and another protein called Rho. And these guys, like tubulin, bind to GTP, and they can hydrolyze GTP. So they go through a cycle of binding and hydrolysis of a nucleotide.
Hmm.
But in this case, we tend to think of them as, to get to one of your questions, basically information molecules. So
Yeah.
we're a pattern of information, and the information that they convey results in the accumulation of actin filaments and myosin around the wound. We can't see that in this movie, but you can see the consequence of their accumulation. The wound is closing.
Hmm. Yeah, and for those, so roughly 80% of the people that consume this will be audio only. I'm looking at this video is playing over and over again. There's a ring, like a literal circle, and this is just fluorescent stuff in the cell that they've engineered, and it's closing up pretty quickly. And there's two colors. Um, we're watching it obviously at one time scale here, Bill. But how fast does this happen in the actual cell?
This movie is about 3 minutes long, so the healing can be very, very fast indeed. And this
Mhm.
is a pretty sizable wound.
Yeah. And so obviously you guys got an experimental apparatus you've set up to do an experiment like this. You can point to your little mini laser gun and burn a hole into the cell. But as you said, our cells are getting damaged all the time. I can imagine that, and correct me if I'm wrong, perhaps if I walk outside for too long and I've got a high UV index, maybe the sun is kind of like a laser that can damage my membrane like this. Maybe there's a physical abrasion. Even though I see a cut, there's probably some cells that are killed, but also some ones that just kind of get punctured a little bit physically, and these holes get fixed and repaired.
Yeah, that's right. So we the examples of damage that people tend to think most about are in mechanically active tissues like muscle. So,
Mm,
and if you're someone who does, say, power lifts or something like that, or even if you're just exercising, your
yeah.
muscle is constantly getting damaged, the and that's normal, okay? But it normally fixes itself. Other examples, the lining of your gut. A lot of foods tend to damage the epithelial cells that line your gut, and if you're healthy, that's not a problem. They heal right back up. You alluded to an abrasion. It is almost impossible to have a tissue abrasion without getting the individual cells of the tissue damaged as well.
Yeah.
Some of them are going to die, but a lot of them survive, and they survive by mechanisms like we're looking at here. So it really is kind of a constant of our life. It's just we're much more in tune, I think, with tissue damage because who hasn't cut themselves or
Right. Right. And naively, when I think about tissue damage, just little cuts and bumps I get in life, I often think like, okay, like I can understand some cells die and then they can be replaced. The gut and the skin have a high turnover rate and they can be replaced. It's easy to forget that not all of the cells need to be replaced. Some of them just get damaged somewhat, but they can be fixed without like a whole new cell needing to come in here. So wound healing happens like at the cellular level itself. Individual cells can become hurt and then better again.
Yeah, that's exactly right. And it's a relatively new process in terms of people getting into its study. But it turns out that the mechanisms involved are fairly ancient. So some of the same players, like Roe, for example, shown here, that participate in the damage response in, say, frog or human or mouse or fly tissues. Sorry, fly cells also participate in the healing response of, say, yeast,
Yeah.
or worms. So,
Yeah. I was actually going to ask you at some point the players involved here, the actin monomers, the Rho, GTPases, this stuff. This is fundamental cell biology here. It's not like this stuff is only in the Xenopus embryo or only in our cells. So I would imagine that there's a lot of evolutionary conservation here, and a lot of the phenomenon that you guys observe in whatever cells we're looking at from the lab here are probably at play like in all kinds of creatures, not even just animals, but plants and microbes and basically whole organisms.
Yeah, that's an excellent point. So there are some old studies, and I think probably newer ones coming along that indicate that plants also can heal individual cells and in an actin-based fashion. It's a little harder to work with some of these organisms like plants because they have cell walls, so making the wounds can be a little more tricky. If you look hard enough at the literature for things like bacteria, prokaryotes, it's pretty clear that they can survive damage as well, which implies that they must have some sort of wound healing response.
Mm-hmm, mm-hmm. Is there anything else that we should look at in terms of the videos that you have on hand here?
The so this sort of it turns out what we're looking at here that certainly they're rings, ring-like patterns, but they're actually waves of activity.
Mm-hmm.
And the idea is that the inner edge of that ring of Rho, it's getting turned on, so it's basically binding to GTP, and then at its outer edge, the GTP is getting hydrolyzed. Same process we were talking about before. Okay, so it's getting turned off, and the same is true for the CDC42. So at its inner edge, it's getting turned on, and its outer edge, it's getting turned off. So what we're actually looking at, believe it or not, are examples of self-organized waves.
Hmm.
There is an upstream stimulus, namely the wound, but these two proteins basically sort out into these wave-like arrangements as a consequence of self-organization.
Mm-hmm, mm-hmm.
The other examples I have here are mostly for those embryo waves and for the waves that split the cell in half, the Rho waves. Also, apparently, dependent heavily on self-organization.
Yeah, yeah. Can we see the one of the cell splitting in half?
Yeah. So here is a cell dividing.
Whoa.
Okay.
Yeah.
So the first thing you notice, obviously, is the cell shape change. It's getting pinched in half and forming two daughter cells. Okay. But we're also looking at, once again, our favorite protein, or one of our favorite proteins, Rho, in cyan, and another favorite protein, actin, in red. So actin filaments. But just for simplicity, we're calling it actin here. And if you look very carefully at this movie, which commences just after the chromosomes start separating, you'll notice that initially there are little flashes of actin and Rho everywhere,
Everywhere. Yeah.
but then they're very rapidly confined and amplified at the equatorial cortex. Okay. So, and that confinement and amplification coincides with the onset of cell division. So we can't see it in this movie, but the mitotic spindle is there. It's what's responsible for separating the chromosomes, but it also concentrates the Rho in these little flashes at the equator. And these little flashes, it turns out, are waves. It's a little harder to
Wow.
see than that wound healing movie, just because even as those waves are forming and moving up and down the long axis of the equator, the furrow is ingressing. So it's moving away from us and away
Mm-hmm.
from microscope objective. So they're only around visually for a short period of time, but we
Yeah.
have
Yeah.
tried to make them last a lot longer.
Yeah.
And
So I mean, for those that are just gonna be listening, right, we're seeing effectively one blob, and it almost looks like it's sparkly, like it's got electricity in it, and it's like the electricity is everywhere, and then all of a sudden, like it goes just to the middle where these two cells are dividing and becoming going from one to two. What what is enabling the reactions that we're kind of looking at here to become confined like this? What pushes them to one side? What is patterning this stuff in this way?
Yeah, the answer to the what's causing the accumulation, the mitotic spindle basically is um, resulting in the redistribution of proteins that activate and inactivate Rho. Okay, so you use the term electricity, which is actually very apropos because we believe this behavior is underlain by a circuit.
Mm,
Okay, a circuit which hinges on Rho, a protein that activates Rho, a protein that inactivates Rho, and then actin itself, the filament that forms the key part of the machinery that's splitting the cell in half. So the idea is these little wavelets of Rho activity are being driven forward because at their front edge, that's where the Rho activator concentrates. Okay. But they're being terminated at their back edge by the Rho inactivator. And the Rho inactivator binds to actin. Okay, so that this may be a few too many verbal steps, but the idea would be where the Rho activity goes up, um, it's promoting the assembly of actin filaments. Okay, so actin starts to grow and concentrates. Okay, so that gives us little Rho wavelets, but then um, the actin is also recruiting a protein that turns the Rho off.
mm.
Okay. So that's one part of the circuit. But then again, at the beginning, we've got the Rho activator, insofar as there is a beginning, but because it's a circuit, it's really hard to say what starts and what ends things. But the Rho activator is going to give us more Rho activity and move the wave along. And in its wake is going to be actin assembly, recruitment of the actin binding protein that inactivates Rho and then thus Rho termination. So that's a cycle that's happening over and over again, and it's self-organized.
I see. So there's an interplay here between an activator and an inactivator, and it's causing these patterns to emerge based on the concentration and the interplay between these activators and inactivators. At least roughly speaking, we might analogize this to the way you get patterned activity in the nervous system. So, like in the nervous system, we talk about neural circuits, which maybe are a little bit easier, more natural to think about, or at least I'm used to thinking about neural circuits. You've got excitatory neurons and inhibitory neurons in the brain. And the way that neuroscientists often start to think about the canonical patterns of activity that emerge in the brain is, well, you've got certain circuit arrangements and there are certain sort of motifs. And one of those that's very common is that the excitatory neurons and the inhibitory neurons are often like coupled together in a particular way. So I'm thinking here of the concept of feed forward inhibition in the nervous system. And an excitatory neuron will excite another neuron. And that's pretty intuitive to people, right? Neurons excite each other. This is how information flows. But oftentimes that excitatory neuron will also in parallel excite an inhibitory neuron that then shuts off that other excitatory neuron. And, without getting into the weeds, this kind of coupling of excitation and inhibition is key for how you get sort of patterns of activity as opposed to just chaos.
Yeah, that we think a lot about the analogy between what we're seeing here in individual cells and the nervous system, both at the level of the action potential, which is also, of course, driven by positive and negative feedback, but then the waves you can get in ensembles of neurons, that their waves. They're not identical to these waves, of course. The mechanisms are very different, but as you've alluded to, the underlying principles are very, very similar. If you can couple positive and negative feedback in the right way, you can drive wave-like behavior. And it turns out in individual cells, you can drive pattern formation.
Mm-hmm.
And
And
that's
so,
what we're here.
yeah. And so a lot of this stuff depends on concentration and depends on localization, I would think. So the cell's presumably not putting all of this stuff everywhere at equal concentration. It's somehow got to be able to get certain things to certain places in certain concentrations. That's going to be, I think, a key ingredient here. Can you just give us just a basic sense for like, how is the cell getting these things, the actin monomers, the GTPases, whatever? How is it like deciding and getting things to certain places so that patterns like this one that we're looking at here of the two cells dividing actually play out this way?
Yeah, so the idea would be for both the Rho and the individual actin monomers, those are assumed to be freely soluble. So they're, bouncing around the cytoplasm and maybe not entirely equal concentrations, but pretty equal everywhere.
Mhm.
So the trick then is going to be to get the things that activate and inactivate the Rho in a particular place. Okay, so the spindle does this in part by concentrating the Rho activator in the region near the equatorial cortex. Okay, so there's simply more of it there. And when there's more of it there, that means even though Rho itself is freely soluble, you're going to start to get more and more active Rho, specifically at the equator. It may also be distributing the inactivator, but it's also possible that the inactivator is just coming along for the ride because it wants to go where the actin and the Rho are. Okay. The actin in turn is going to end up getting concentrated at the equator due to the action of the Rho. So when the Rho is turned on, it binds to proteins that or promote actin assembly. So even though actin does have the capacity to self-assemble, these proteins make it more efficient, basically.
Mhm. So you can have certain components of the cell soup can basically just kind of go everywhere. They're just kind of like diffusing around, just like, sugar and water if you were to mix it together in a cup. But then other things like some, certain proteins or enzymes or whatever, they can kind of get put in particular places, but then because some reaction is happening that then changes the stuff that's everywhere in roughly equal concentration, it starts to change. And then obviously you're going to get patterns that emerge based on these reactions starting in certain places based on the concentration and the localization of proteins. And I guess this starts to automatically, in my mind at least, explain why we talk about emergent phenomenon. It's like, oh, okay, one component of the equation here can kind of go everywhere. If we localize another component and then from one point in the cell a reaction starts, that's naturally going to travel. I mean, it's pretty intuitive to me. That's going to result in kind of waves of stuff happening.
Yeah, that's exactly right. And it's a very clever thing for evolution to have landed on because you can generate diverse patterns, you can make them flexible. So imagine, for example, that the spindle has moved to one end of the cell or the other, or been pushed for whatever reason. And this commonly happens in tissues. You really still want the furrow to split the cell in half between the separating chromosomes. So you don't want to have a top-down decision-making mechanism that just says split in this place where the chromosomes were that corresponds initially to the midplane of the cell. You need to be flexible in case the spindle shifts. This way, doing it this way makes sure that the cell can actually respond to changes in the spindle position.
Mm-hmm, mm-hmm.
Same is true for the wound healing response. You want the cell to be able to respond where it's damaged, not obviously, where it's not damaged. So that means the machinery has to be flexible. It has to be able to respond quickly. And doing it this way via self-organization makes that possible.
When we talk about cellular level wound healing, like the last movie we were looking at, and we're thinking about actin and the cortex and the plugging up of holes, right? The cell gets wounded, like a physical hole gets burned into it in some cases by Bement because you have a laser gun in your lab. And then this actin magic happens and the wound heals. And it's amazing. You mentioned, correct me if I'm wrong about any of the details here. I think you said that each actin monomer is associated or can be associated with an ATP.
Yeah, that's right.
So I would imagine that when these cell cortical rearrangements are happening, when holes are getting plugged up, these actin filaments are probably pretty damn long if in terms of the number of monomers. This isn't probably just one or two. I'm guessing they get pretty large. What I'm getting to here is how energy intensive are these cytoskeletal rearrangements? My guess is they require a fair amount of ATP.
Yeah, but so here's the thing. There isn't a huge literature on this, but when people have made the effort to try and track the energetic investment in various cell costs, the big ticket item always seems to be protein synthesis, and especially the synthesis and insertion of proteins in membranes.
Hmm.
And I think it would definitely be true if you popped a number of wounds in a cell, if you just kept wounding it over and over, eventually you're going to seriously impact the available ATP. But I think most people who study where the energy goes would argue that that's still going to be not as much as you would imagine compared to the background processes needed to just maintain the cell in its steady state.
Okay, so normal. So in other words, normal healthy cell, a normal cell, which means from time to time it's going to take on like bits of damage here and there, but not catastrophic. Nothing out of the ordinary for run of the mill cell doing its thing. You're saying that things like protein synthesis and the regulation of insertion of proteins into the membrane, those things are generally thought to be more energy intensive than the stuff we're talking about here.
Yeah, now with a caveat that that claim is rests on just a couple of papers, and
Right.
it's the sort of thing it takes a lot of effort to do really well and carefully. So I think in general, uh, I certainly couldn't claim that people have measured the energetic cost of healing. That has
Yeah.
not been it's more when they sit down and calculate the known amount of energy required to make a single protein and shove it into a membrane through a pore, versus the estimated amount of energy from other sources. It's the synthesis and things like that that are apparently very, very expensive energetically.
Yeah, yeah. Okay, so that's the thinking, but like there's a lot of question marks here because it's
Yeah.
probably really hard to quantify precisely how much ATP all of these individual things actually use up.
And it's a really important question from the standpoint of something we were just talking about, which is that self-organization requires energy input.
Hmm.
So even though it doesn't take ATP hydrolysis to make an actin filament per se, it does take the hydrolysis for the example, for the filament to disassemble. It takes the hydrolysis to make one end of the filament different from the other. So to get a properly self-organized structure like we're looking at here, or we're previously looking at for the wound healing, there, there's probably energetic investment in multiple steps, and it would be wonderful if it were possible to start teasing those steps out because getting back to the second law of thermodynamics again, where a pattern basically represents order, which means
Yeah.
we have to be investing energy to make it happen.
Yeah, because the second law tells us that spontaneously we should get more disorder with time. And yet, obviously, when we're talking about life and cells, like all of these patterns are clearly emerging. So some kind of energy has to be coming in to drive that.
Yeah, and you've touched on a really key question, I think. Where does the energy go? And suppose you wanted to make your own cortex artificially, like
Hmm.
probably you'd have to come up with the means to invest energy into it to give it all the capacities that a real cell cortex has. So how do you go about that? Well, first thing you want to know is where the energy normally goes, I think. So I, I do think that it's going to happen. Eventually, people will start figuring this out, but it's far from trivial.
Hmm. I want to talk about a couple of things, and I'm not sure how directly this will tie into your work, but I just want to see where we go because some interesting things are coming to mind here that potentially tie some threads together that cut across a bunch of the episodes I've done. I'm gonna ask you about cancer and wound healing in a little bit, but first I want to talk about when wounds happen to the membrane. Obviously, in the lab, you can do experiments like the one you showed us before, where you can make a wound on purpose by firing a laser at the membrane. But the membrane is obviously an important part of the cell, as we've discussed. It can be damaged, obviously. What are some of the natural ways that cell membranes become damaged in a normal biological situation in an organism? I'm thinking of things here like, okay, when we if we understand metabolism at a basic level, we know that in oxygen-based organisms like ourselves, right, we always have to deal with some level of reactive oxygen species, which can then react with components of the cell membrane. And people often analogize this to, like a little spark causing a fire somewhere. So when I think about the cell membrane of an oxygen-based organism, an aerobic organism like an animal, what are some of the ways the cell membrane tends to or can be damaged naturally?
Yeah, there are a lot. That's the short answer. So the definitely, chemical insults, inappropriate oxidation, that can cause problems. The scale of the wound will vary depending on the insults. So some of the smaller things like chemical damage, those can probably be handled by the cell without the sort of extreme sort of response we were seeing before. You'd probably need to, remove the damaged membranes. And sorry, the damaged lipids and then replace them. But there are other things that are a little more severe. So certain bacteria come with toxins that poke holes in membranes, and there you're going to need a response. It may or may not be as extensive as the one we are looking at, but you have to stop the calcium from rushing into the cell because it will ultimately kill the cell if it's
Mhm.
allowed to. So if your bacterial toxin has plopped some pores into the membrane, the cell is going to do things like try and internalize those pores and get rid of them. It will typically shove a whole bunch of membrane into the site of the pore. There's a general response that wouldn't have been evident in the movies we were just seeing because we weren't using the probes that let you visualize the membranes. But besides the sort of accumulation and closure of cytoskeletal material around wounds, there's also picture an accumulation of membranous compartments
Mhm.
on the interior of the wound. They can be cross-linked to each other by certain proteins to sort of plug the hole. So that sort of thing seems to happen even with the smaller wounds.
Yeah,
And
yeah. Bill,
so,
can you actually can you stop screen sharing now?
oh yeah, sorry.
One of the things I've been very interested in and studying quite a bit the last couple of years is lipid peroxidation. So when we think about fatty acids in cells, they can be oxidized on purpose, so to speak, through enzymes when the cell wants to use them to make energy. And they can also be structural components of membranes. So the phospholipid bilayer of the membranes that we've been talking about there's a bunch of different fatty acids in there. They can be saturated, monounsaturated, polyunsaturated. So there's different versions of these. And one of the phenomenon I've been talking to people quite a bit about recently is lipid peroxidation. So you might have, say, a reactive oxygen species gets spit out of a mitochondria or something, and then it reacts with the membrane. And depending on the specific type of fatty acid it reacts to, different things can happen. And in general, polyunsaturated fatty acids can be oxidized by reactive oxygen species more readily than other fatty acids, and then they can produce toxic molecules. And there's this notion that when you change the fatty acid composition of the membrane such that you've got more polyunsaturated fatty acids, and especially if there's a bunch of them in high concentration next to each other, you can get these like chain reactions that happen. And so you can almost imagine like a little hole getting burned in the membrane if you've got a bunch of fatty acids that get, lit on fire, so to speak, by a reactive oxygen species. Is there anything that you can say or anything that you know about like the fatty acid composition of the membrane and how that might relate to the propensity for wounds to arise in the membrane and the cell's capacity to handle them?
Yeah, I can't comment specifically, but it would be remarkable if that didn't come with some kind of consequences for wound healing.
Mm-hmm.
And there's a sort of complex interplay between cell aging and cell healing. So from the standpoint that excessive cell damage tends to shorten the lifespan of cells. This is very recent work, not ours, Keiko Kono's work. But then there's also reason to think that as cells get older, they're crappier at healing, if I could put it that way.
Mm-hmm.
And I would imagine if you're altering the lipid bilayer makeup, that is, it almost has to be the case that it's going to render the cell basically less able to heal wounds because the membrane damage comes with a characteristic. or elicits a characteristic reorganization of membranes around wounds. And actually, it's also a concentric pattern similar to what we were seeing for the Rho and the CDC42, but even more extensive. So and it seems to be important for the healing. So if you're altering the makeup of the plasma membrane lipids, there's an excellent chance that that part of the response is going to be compromised. I just And then as well, the most cells are constantly subjected to mechanical stress. So it's extreme in the case of muscle, but picture the lining of your gut. We talked about that a little bit before, but food is passing down it. There's peristalsis moving the food. Okay, the peristalsis comes with force imposition. Blood flow comes with force imposition. So all of that has the potential to cause small damage to the cells in question, enterocytes for the gut, um, endothelial cells for the blood vessel. So once you start, directly or indirectly changing the composition of the membrane, that means in all likelihood the physical response to the damage is going to be more severe. I could be wrong, but I just have a hard time. Things just don't tend to work better when you start changing their normal makeup.
Yeah, yeah,
At
yeah.
least,
I mean, what this plugs into in terms of a lot of the other content I've done is that the dietary fatty acid compositions of humans today and for X number of decades now is basically unlike anything we've seen in human history and prehistory and basically in the animal kingdom. And so the implication is that if you change the fatty acid composition of membranes and things in the cells, this has to change them in some capacity in terms of what they're vulnerable to. And lipid peroxidation of PUFAs is something people talk about a lot because they are more readily oxidized by reactive oxygen species. They do produce toxins. And I was just trying to fit this together with what you were showing me about wounds. It's very easy for me to imagine, if you get a sufficient density of, say, polyunsaturated fats in the membrane, it's going to be really easy for that hole to burn bigger than it otherwise would.
yeah, I don't. So this is not exactly on point, but I think it supports that line of thinking. So this is work done by Paul McNeil a few years ago, but they showed that there's a link between diabetes and impaired cell repair.
Mm-hmm.
And they went further than that. They showed that they could actually take cells and give them an excess of glucose, just isolated cells. Okay. So in other words, mimic essentially a dietary condition, although of course, glucose imbalances can arise from a number of different ways. But in one of their experiments, they're actually adding, glucose to the medium. And it turns out this also eventually resulted in repair deficits in the cells exposed to this manipulation. So I would argue that at least in that case, there's a clear connection between things we think of as food intake and in particular deleterious food intake and what's happening to the cell repair response.
Mm-hmm. Mm-hmm. And other conversations I've had around thinking about cancer not so much as a DNA mutation driven thing, but more of as a broken metabolism driven thing. Apparently this goes back quite a long time to a man named Virchow who hypothesized that cancer itself kind of represents a deficit in wound healing at the cellular level. Like something is going wrong with how the cell is using energy and it can't repair its wounds at all. I'll let you take this anywhere you might be able to take it, but how does the work that you do on cellular wound healing tie into or have implications for things like cancer biology?
So I will say we tend to more think about diseases like muscular dystrophy, but I can point you to
Mm-hmm.
a really interesting set of experiments. It's related to cancer and cell, um, but not quite in the sense that I think Virchow meant it. But this work was done by Jody Jeswal, and what he found was that highly metastatic cells, the ones that are really deadly during cancer, the ones that, get disseminated away from the initial cancer site. This behavior seems to be associated with an upregulated capacity for self-repair.
Hmm.
And this initially doesn't seem very intuitive. Like, well, why would that lend some kind of advantage to the cancer cell? It turns out to be fully metastatic. A lot of the cancer cells have to squeeze their way through other tissues, maybe force themselves
Mm.
through the lining of the blood vessels or whatever. Normal cells always, in the consequence or as a consequence of doing this, which and it can happen naturally, they often get damaged. This is another mechanical stress. Like they can actually have a chunk of the plasma membrane torn out, apparently. The cancer cells are really, really good at healing the torn out chunk of cells because
Mm.
overexpress a couple of proteins, or at least one protein involved in the healing response.
So the
So
idea sounds like it's not just so you've got metastasis, some cells become metastatic, they can move around from the original site, but as a consequence of doing that, you're then exposing yourself as that cell to more potential damage because you've got to move and squeeze through crevices and stuff. And so, in order to accommodate that and actually get somewhere and stabilize, and continue growing, they've got to be better at repair.
yeah, exactly.
Wow.
And the data were really, really impressive. And it's sort of like, oh wow, to be honest, it never occurred to me that that kind of repair would matter for a metastatic cell, but apparently it does.
Wow, wow. So what are some of the things that you're working on in the lab today? Where's the cutting edge when it comes to the cortex and cytoskeletal dynamics?
Yeah, we're trying to understand exactly how this self-organization works. We talked about briefly a circuit. The circuit is apparently more complicated. There are more loops, which is not won't be a huge surprise to you, right? The nervous system, it's not like it's like two neurons talking to each other. It's like there's a whole network. So
Hmm.
same
Hmm.
sort of thing, but transplant it into the interior of a single cell. So the various players are talking to other things that then interface with other feedback loops. So we're trying to pin those down and trying to figure out what they bring to the table. What do you get out of having more feedback loops? The cell, the system, and that's apparently true for both the wound healing and the cell division. Um, we'd like to learn enough about these behaviors to be able to manipulate them. Of course, it would be wonderful to be able to develop or help people develop strategies for overcoming deficits in cell repair, for example. The the muscular dystrophies remain a very, very serious and relatively common set of
Hmm.
diseases, in spite of a lot of effort to help people out.
Can you give us
So
just a very, very basic explanation of what the cellular problem is in muscular dystrophy? Why can't the muscles fix
yeah,
themselves?
so take this with a grain of salt, because this is coming not from a physician standpoint. It's coming
Hmm.
from basic cell biology standpoint. But I feel like you can sort of distill the issues into two. So in the more deadly example, Duchenne's muscular dystrophy and related muscular dystrophies, the process of muscle contraction produces wounds that are much larger than normal. And that's due to improper coupling between the contractile machinery of the muscle and the muscle plasma
Hmm.
membrane. So
So every time the muscles contract, more damage is happening than would otherwise.
yeah, it's just worse than it would be for a normal patient. And eventually it simply outstrips the repair machinery's ability to fix the
Hmm.
damage.
Hmm.
That's one way to
So
think of it.
it's not a deficit in repair. It's too much damage happening.
Yeah, for that particular kind. But there's another class of muscular dystrophies, the so-called limb girdle, and many of those seem to be underlain by deficits in the repair process. So they're the mechanical or the coupling of the contraction to the muscle plasma membrane, or sarcolemma as it's known, seems to be normal. But when the damage occurs, even though it's normal, the repair systems aren't functioning properly. So the end result is the same. You end up with muscle atrophy just because the holes can't be fixed. And then this all comes with a lot of other events that of course are bad for the patient. But that's one way to sort of sort the two kinds of dystrophy out.
Hmm. Hmm. And so what other kinds of things are you working on in the lab today?
Um, we're also trying to figure out the machinery that makes a cell split in half and how that self-organization works. And one of the more interesting things we're doing is we're trying to develop a set of tools for synthetic self-organization. So basically, we're stitching proteins together that we think mimic or are designed to mimic the natural ones that drive self-organization, with the goal of figuring out what the key parts are. So it's one thing to say, oh, here's a row activator. Well, how does it work? What does it take to give you the positive feedback? What are the other add-ons that it comes with that really make the system work properly? And that's turning out to be really interesting too. I don't,
Hmm.
it's all interesting, but
Yeah, yeah, yeah. One question I'll just ask you for fun. I know this isn't what you're literally working on, but when we think about self-organization, the work that you do, despite your focus on certain particular forms of all this stuff, as we start to distill down and learn what like the principles of self-organization are in biology, I mean, to me at least, right? This naturally then brings you to questions around like the origin of life. Like how did a soup of stuff, like in a volcano or whatever, at some point self-organize into what became cells that then, became us ultimately? What's your general take on like when you think about the chemistry, the biology, what we know and where we're at with self-organization and the technologies that are out there that allow you to do the stuff that you showed us? How close, what's your gut tell you about how close we are to creating life in the lab? Is that 100 years from now? Is that within our lifetime? What do you think?
it would probably depend on how you're going to define life in the lab. I know
Hmm.
some people would argue that if you make a self-replicating anything, that's life. But the usual laundry list of definitions of what comes with life is quite a bit longer than self-replication. So I think it's still a ways off, but I do tend to feel, after learning about and seeing cells actually execute some of this self-organization, that again, as we were talking about before, it might not be as hard as I always assumed. If you can get, just by having a couple of proteins, an activator and an inhibitor, if you can get these complicated patterns forming, some of the stuff that just looks really deeply complicated and mysterious might not be as complicated and mysterious as we thought. So
Yeah.
I argue that it might be closer than we think.
Yeah, yeah, yeah. Remind me, who did the experiments where they put DNA on a bead and you basically get the
Yeah,
chromosomes lining up?
that's Rebecca Hild. She's at the University of California at Berkeley, and this was, I think, during her postdoc work. But it's since that time, people have done all sorts of things with extracts and just having a cell soup, as you put it, is really convenient, and it can reveal all sorts of what we previously thought were just impossibly complicated things that can happen without, the nucleus or whatever telling or providing the directions. It just happens as a consequence of things bouncing around long enough and at the right concentration.
Yeah, yeah. Are there any final thoughts you want to leave people with? Anything you want to reiterate about your work, about cell biology, or some of the self-organization phenomena that we talked about?
Um, there's an old joke that never ask a barber if you need a haircut. I feel like, that aside, I want to give a pitch for cell biology. I think there's just that people have this tendency to look at a big fat textbook and, oh yeah, we know all about cells. That I don't think we know that much. I think people should just spend more time looking at them. A lot of this stuff comes out of simply filming their behavior. And if I were king of the world, I would maybe set money aside for people to, or more money aside. I said, yeah. Anyway, the
Yeah, yeah, yeah. So you're just saying we don't know as much as we might like to think, and we might suppose we do when we crack open a textbook in cell biology just because it's got 1,000 pages. And I think what you tried to say there was you can learn a lot just by watching videos of these things. And we haven't been able to really look at good videos for that long because of technological constraints. And there's still much to be learned. And we might be surprised at how simply the observations can be by watching these movies in terms of what you can discover.
Yeah, that's exactly the point I was trying to make. Yeah.
And yeah, so for those who are listening, there were some cool videos we looked at that'll be on the video version. But if that's it, Bill, thank you very much. I still remember taking your class. It was a fun class. I didn't want to take it. I had to take it. It was like one of those where like I was like, hopefully this is okay. And it was a really cool class, and I still remember it.
Well, thanks for that. I do think the cells are just fascinating, but okay, well, thanks for having me on. This was fun.
Visit mindandmatter.substack.com to find all of my content. If you like to read like I do, then you should check out the ReadWise app. It's totally free to download, and if you go through the link in the episode description or on my support page, you can get a free 60-day trial for zero cost. ReadWise is a really cool app that allows you to import digital highlights from over 30 different platforms such as Kindle, iBooks, PDFs, and more. It makes it really easy for you to review and find and share excerpts and quotes from whatever it is that you're reading, whether it's PDFs of scientific papers or books that you're reading on your Kindle. And you can even save and use highlights from physical books. I read a lot of physical books before bed, for example, or when I'm traveling. And the ReadWise app actually integrates with the camera on your phone so that you can take a photo of a page that you're reading, highlight a specific chunk of text from that page, and export it to the app so that you can reference it or find it or share it later. They even allow you to export really cool looking graphics of some of the highlights that you have in the books or the papers you're reading so that you can share them in an engaging way. So check out the ReadWise app today if you are a reader and you like to reference and organize what you read. Go through the link in the episode description or on my support page or in my free weekly newsletter at mindandmatter.substack.com and try the ReadWise app 60 days free today.